Laura Piccio, and today I will talk about our studies on the effects of calorie restriction in MS and its main animal model, experimental autoimmune encephalomyelitis, EAE. During this presentation, I will first summarize the studies showing the association between obesity early in life and increased risk of developing MS later on. Next, I will discuss some of the potential mechanisms underlying this association, such as the role of the adipokines, which are cytokines produced by the adipose tissue and the gut microbiota. And finally, I will present our studies on calorie restriction in the MS animal model and in people with MS. So we still do not know the cause of MS, but we know that it's a multifactorial disease due to the interplay of genetic and environmental factors that, through mechanisms that are still for the most part unknown, are leading to an autoimmune response against CNS components. In regard of the environmental factors linked to MS risk, several epidemiological studies in the past 10 years have shown that obesity in children and adolescents is associated with increased risk to develop MS later on in life. Other environmental factors that are considered to be potentially important are diet and gut microbiota, and these are currently under intense investigation in preclinical models, but also in people with MS. So what do we know about obesity in MS? Several studies have looked at an association between established MS and obesity, with results that are not consistent among the different reports. In contrast, numerous epidemiological studies have consistently shown that being obese or overweight during childhood, adolescence, or in young adults is associated with increased risk of MS, suggesting that the metabolic state associated with obesity in this phase of life could set up the stage for developing autoimmunity later on. On the other hand, prospective studies confirm the association between body mass index and MS activity and progression, and specifically higher body mass index were associated with increased risk of relapse, as well as increased disability and brain tissue loss at the MRI. So the first study that reported an association between obesity in adolescents and risk of MS was published in 2009. It was a longitudinal study that used two large cohorts of American women, the Nurses Health Study 1 and 2. They reported a twofold increased risk to develop MS among women with a BMI higher than 30 at the age of 18. This finding was replicated in a second study in Sweden. This was a case-control study confirming this association and extending the observation also to males. Subject with a BMI higher than 27 at the age of 20 had a more than twofold increased risk to develop MS compared to subjects that were normal weight. Then two other studies were published reporting in two different pediatric populations that childhood obesity was also associated with a significant increased risk of MS. And next, these findings were confirmed in several other reports. However, these are all epidemiological studies which are reporting an association between obesity and MS risk, and the main limitation of these epidemiological studies is that they not necessarily demonstrate a direct cause-effect relationship. However, more recently, these findings were also confirmed in Mendelian randomization studies, suggesting that there could be a real causal relationship between increased BMI in children and MS development. The mechanisms underlying this association are likely to be very complex, and they are not exactly known. Some of them are probably related to changes in the adipose tissue and in the production of cytokines that are produced by the adipose tissue and also the gut microbiota, which is what I'm going to discuss now. So in fact, we know that the adipose tissue is not just a fat storage, but it's viewed as an endocrine organ, which is capable of secreting hormones and cytokines, which are collectively referred as adipokines. Adipokines regulate metabolic functions, but in many cases they can also have direct effect on immune cells of the adaptive and innate immune system. Therefore, they can create a link between metabolism and immune inflammatory responses. Certain adipokines can have an overall anti-inflammatory action, like adiponectin, while others can have an overall pro-inflammatory activity, like leptin. Obesity is characterized by increased adiposity, and also by the infiltration of the fat with immune cells such as macrophages and T cells, which can also be the source of adipokines. In obesity, the total amount, but also the proportion of the different adipokines is altered. And this eventually has important metabolic effects as well as effects on immune and inflammatory responses. In this regard, it's well established that obesity is characterized by the release of cytokines, adipokines, shifting the balance to a more pro-inflammatory milieu, which leads to a chronic, low-grade inflammatory state that may promote autoimmunity. So this is likely a central mechanism linking obesity with increased risk of autoimmunity. However, there might be other potential mechanisms, including the fact that obesity is associated with lower levels of vitamin D metabolites. These molecules are lipophilic molecules that can get trapped into the fat. That's why circulating levels in obese individuals are lower. And vitamin D we know has an important immunomodulatory effect. Another factor could be the role of the gut microbiome, which is, we know it's altered in obesity with higher proportion of pro-inflammatory bacteria. So the gut microbiota, as many of you probably know, are the communities of commensal microbes that we host in our gut. While in the gut mucosa, there are numerous resident immune cells, including lymphocytes, dendritic cells, macrophages, with the primary protective role against potential threats from the environment. There are close interactions between these two compartments, and the type of microbiota has a strong influence on the type and function of immune cells that are associated to the gut. The gut microbiota can modulate immune responses directly through bacterial components that can directly interact with the immune cells, or through the production of metabolites that can have immunomodulatory functions, such as the production by certain bacteria of short-chain fatty acids, which are the results of the fermentation of dietary fiber, and we know they can have important immunomodulatory effect. What is important is that changes in gut microbiota and gut immunity could also have systemic effects on the immune system. The situation which is at the other end of the spectrum compared to obesity is calorie restriction, and several lines of evidence suggest that calorie restriction, in short CR, is associated with the reduction of systemic inflammation. And the mechanism could be the opposite of what we just discussed for obesity, with an altered production of adipokines by the adipose tissue that would favor towards a more anti-inflammatory milieu. So this is essentially the main rationale of our studies, and why we decided to test the effects of calorie restriction first in the EAE model and then potentially in people with multiple sclerosis. Calorie restriction is defined as a chronic or intermittent reduction of food intake below usual ad libitum intake without malnutrition. Calorie restriction can be implemented as a chronic or intermittent approach. In chronic calorie restriction, food intake is reduced every day, while meal frequency is unchanged. And usually in preclinical model, people are able to test from 20% up to 40–50% of chronic calorie restriction that can be achieved in animals. While in humans, usually it's around 20–25% that can be done. In intermittent calorie restriction, we have several potential approaches. In preclinical model, usually intermittent calorie restriction is usually intended as alternate day fasting, which consists in cycles of 24 hours of fasting and feeding. While in clinical studies in humans, intermittent CR can be done with in different ways. Can be intermittent fasting, in which people are usually fasting every other day, and it can be water fasting every other day, or 2 or 3 days per week in what is called the 5-2, which commonly called as the 5-2 diet. Or it can be not necessarily complete fasting, water fasting, but it can be a significant reduction of food intake, and in this case every other day or 2 or 3 days per week, and this is more properly defined as intermittent energy restriction. And finally, another possible approach is time-restricted feeding, when the food consumption is limited into a 6–8 hours window during the day, while usually it's fasting the remainder of the day. As I mentioned before, calorie restriction is known to have an overall anti-inflammatory effect through different mechanisms, including those that we discussed, reduction of adiposity and changes in adipokines levels, as well as effects on the gut microbiome. But there are several more adaptation, metabolic and hormonal adaptation that are induced by CR that can lead to a reduction of systemic inflammation, including, for instance, an increase in the parasympathetic tone, which has an anti-inflammatory effect, as well as an increase in the production of glucocorticoid hormones, which are potent anti-inflammatory molecules, or effects on the reduction of senescent cells, which are also pro-inflammatory, and a reduction of glucose and N-proxo that can so reduce also inflammation. So based on this rationale, as I said, we decided to test the effects of calorie restriction in the main animal model of MS, which is EAE. EAE is induced in mice after immunization with myelin antigens. Clinically, it develops as an ascending paralysis that we score clinically using a scale from 1 to 5. And even if EAE does not reproduce all the complex aspects of the human disease, it has been instrumental to give some insights about the pathogenesis of the disease in humans, as well as to develop several of the current treatments for relapsing MS, or at least it has been shown that all the medication that did not directly derive from the EAE study, they could also be effective in EAE. So it has had some utilities. So before I go into our CR studies, I would like to give a brief overview of those evidence highlighting the relevance of adipokines at gut microbiota in the EAE model and potentially in MS pathophysiology. So the two main adipokines produced by the adipose tissue are leptin and adiponectin. The role of both of them has been investigated in MS preclinical and clinical studies. Specifically, leptin regulates, has a primary metabolic role to regulate food intake and energy expenditures. Its levels are directly correlated with the amount of body fat, and leptin is known to have an overall pro-inflammatory effect by stimulating both adaptive and innate immune system. So leptin is important to fight infection, but if abnormally elevated, it could promote autoimmunity. According to this view, leptin knockout mice are resistant to EAE, and they gain susceptibility when leptin is administered through mechanisms that are mediated also in part by regulatory T cells. Adiponectin, on the other hand, is an anti-inflammatory molecule, as a primary metabolic role is an insulin sensitizing molecules, and we have demonstrated that adiponectin knockout mice, they develop a more severe disease, while administration of adiponectin in regular B6 mice is able to ameliorate the disease, again through modulation also of the number and function of regulatory T cells. Leptin and adiponectin have also been studied in people with MS. Leptin was found to be upregulated in active MS lesion, and its levels are reported to be higher in the blood and CSF in people with MS compared to healthy control. It was shown that there is an inverse correlation between leptin levels in blood and circulating regulatory T cells, as well as that an increase in leptin was associated with an elevated MS risk in young individuals. Adiponectin also was studied in people with MS, and the results of adiponectin levels in blood have been studied, by different groups with not consistent results, while the twin study showed that CSF levels of adiponectin were higher in the affected twin compared to the unaffected twins, and higher levels of adiponectin were associated with lower risk of relapse in pediatric MS patients. There are numerous evidence also of the role of the immunomodulatory role of the gut microbiota in EAE. So we know that germ-free mice do not develop EAE, and similarly treatment with antibiotics, which reduces the load of gut bacteria, is protective in EAE. Germ-free mice, they gain susceptibility once their gut is colonized with a certain type of bacteria, the segmented filamentous bacteria, which promote the differentiation of IL-17 T cells and EAE development. And on the other hand, treatment with polysaccharide A, which is a component of the human commensal bacteria, Bacteroides fragilis, ameliorate EAE through a mechanism mediated by regulatory T cells. And we also know that dietary factors can modulate EAE, potentially through a mechanism mediated by the gut microbiota. The gut microbiota has also been studied in people with MS compared to control in a number of studies. Overall, these studies have shown modest patterns of gut dysbiosis, and what was shown was mainly differences in single taxonomic units and not really large-scale changes in the structure of the gut microbiota when the gut microbiota in people with MS was compared to healthy control. In most cases, there is not a lot of consistency, but there are certain taxa that were noted to be differentially represented in several studies, including, for instance, an increase in Akkermansia in MS patients, as well as a decrease in Faecalibacterium, Clostridium species, and Prevotella strains, which are more related to an immunomodulatory phenotype. Whether these gut microbiota alteration in MS could contribute to disease pathogenesis or are just a consequence remains unknown, but in support of the real pathogenic role is the finding that the transfer of gut microbiota from people with MS can worsen EAE compared to the transfer of gut microbiota into mice, compared to the transfer of gut microbiota from healthy control individuals. And now I will present our main results. So we have studied the effect of calorie restriction in the EAE model by using two different approaches. In initial experiments, we have done 40% chronic calorie restriction. Mice were eating 40% less of what they would have eaten ad libitum for 4 weeks, and then they were immunized to induce EAE. As you can see from these graphs, the red line are the mice that were on calorie restriction compared to the ones fed ad libitum, the black line. On the x-axis is time after immunization, on the y-axis is the severity of the disease, and you can see that chronic calorie restriction was effective in ameliorating the disease in two different strains of mice. So we observed a delayed onset, decreased severity, and also a decreased incidence. However, we figured out that chronic calorie restriction would be very difficult to do in humans, and so we switched to a different protocol, intermittent fasting. But before we moved into people, we wanted to test also the effect of intermittent fasting in the EAE model. So intermittent fasting in these experiments were done as every other day fasting. So the mice, they were going through 24-hour cycles of fasting and feeding. This intervention was done for 4 weeks, and then mice were immunized, and we followed the EAE clinical course. And as you can see from this graph, again, we saw a pretty significant effect of intermittent fasting in ameliorating disease clinical score. So during this, and from now on, what I will present are mainly results obtained in the experiment in which we tested intermittent fasting. We obtained serum and stool samples at different time points, which are indicated at T1, T2, and T3. T1 was baseline, T2 it's after 4 weeks on the diet before immunization, and T3 during clinical EAE, in order to better understand the effects of this diet intervention on the disease. First, we evaluated serum levels of various mediators that are involved in the regulation of inflammation. So we measure serum levels of corticosterone, which is the principal glucocorticoid hormone in mice with a potent anti-inflammatory activity. As you can see, corticosterone levels were not significantly different between the two groups at baseline, while after 4 weeks of intermittent fasting, levels of corticosterone were significantly higher in the fasting group compared to the ad libitum fed group, and these levels were maintained also during EAE. Next, we also measured serum level of leptin and adiponectin. Again, at baseline, there were no difference in levels of leptin and adiponectin between the two groups, while after 4 weeks, we observed a significant reduction of the pro-inflammatory leptin in the intermittent fasting, and a significant increase of the anti-inflammatory adiponectin in the intermittent fasting group. So in summary, intermittent fasting was associated with changes in corticosterone, leptin, and adiponectin that were all favoring a reduction of systemic inflammation. Next, we studied the effect of intermittent fasting on the gut microbiome at the 3 time points, baseline, after 4 weeks, and during EAE. First, we looked at gut microbiome diversity, which reflects the complexity of the microbial community, with higher diversity being associated with a healthier gut microbiome. And intermittent fasting was significantly increasing the diversity in the intermittent fasting group at the T2 and T3 time, while in the control group, gut bacteria diversity did not change over the course of the experiment. It's also known that the gut microbiota can modulate systemic metabolic responses in the host, including levels of leptin. And interesting, we observed by considering both groups, fasting and control, that levels of leptin were inversely correlated with the gut microbiome diversity, which is consistent with what is observed in the literature. Next, we also looked at the immune cells. Since we were able to induce changes in the gut microbiota, we also looked at changes in the immune cells associated with the gut. And we observed that intermittent fasting was inducing a reduction in the proportion of IL-17 producing T cells compared to the control group. These are pathogenic T cells in EAE, as well as intermittent fasting was associated with an increased proportion of Tregs, which have a protective role. We also looked at the effects of intermittent fasting on gut microbiome composition, and we observed an enrichment of bacteria that belongs to the families, for instance, of lactobacillus and provotellacea, which have been associated with a more immunomodulatory phenotype. We have also found direct and inverse correlation between the relative abundance of certain families with leptin serum level to support a dual, a bidirectional interaction between the gut microbiota and host metabolism. Next, to test the hypothesis that intermittent fasting induced amelioration of EAE could have been, at least in part, mediated by changes in a gut microbiota, we performed fecal microbiota transplantation experiment. So basically, fecal matter from mice on intermittent fasting or fed ad libitum were transferred into antibiotic-treated recipient mice. The treatment with the antibiotic was done to wipe out their gut microbiota and recolonize with either gut microbiota for mice that were intermittent fasting or fed ad libitum. And then these recipient mice, they were immunized to induce EAE. And as you can see, we also observed that the disease severity was significantly reduced, the red line, in mice that received the gut microbiota from mice that were on intermittent fasting compared to those that received the gut microbiota from mice that were on an ad libitum diet. So these results show that the gut microbiota from mice on intermittent fasting can transfer protection in EAE. So it suggests that the effects of intermittent fasting could be at least partially also mediated by changes in the gut microbiota. So based on these results in the preclinical model, we decided to move into humans because that is our main interest, and we performed a randomized pilot study in subjects with relapsing remitting MS that were enrolled in this study at the time when they were having a relapse and they were treated with steroids. This study was a small study. We enrolled 16 people with relapsing remitting MS, and the participants, they were randomized to one of two groups, one control group in which they were treated with steroids and they were staying on their regular diet, or one group that was doing intermittent. They were treated with steroids and they were doing intermittent fasting for 15 days, which was every other day fasting, intended as more intermittent energy restriction, because they were allowed to eat small amount of calories around it, which consisted in 1 or 2 salads with the non-starchy vegetable and the light dressing for a total of 400-500. And this was done every other day for 15 days. The participants, they were evaluated at day 0 before starting the steroid treatment and after 15 days. And we were collecting blood, stool sample, and we were doing a clinical evaluation. The primary outcome was the effect of the intervention of the serum adipokine level, and we had also a number of secondary outcomes, including immunophenotyping of blood cells by flow cytometry, studying regulatory T cell function in vitro, and gut microbiome analysis. These are the main results for the serum metabolites in the study. So we focused on those that were altered by fasting in the EAE study. In the graphs on the bottom left, you can see the levels of leptin and adiponectin in the two groups on day 15 that were adjusted for baseline levels and BMI. We observed, as expected, a significant reduction of BMI in the group that was doing fasting compared to the control group, and we also observed a significant reduction on day 15 of leptin. Actually, there was a decrease in leptin in both groups, but the decrease was significantly greater in the fasting group after adjustment for baseline level and BMI. While we did not see a significant difference in level in adiponectin, as well as we did not see significant difference in level of beta hydroxybutyrate, which is the main ketone body in the serum. This is actually was not expected because it's known that it needs multiple consecutive days of low carbohydrate intake in order for the ketone bodies to increase in humans. We also looked at the gut microbiota, and we did not see significant particular changes between the two groups, and this is also expected given the small sample size of this study, as well as the high variability in humans. But we observed a trend towards an increase of the Faecalibacterium, which I said, is known to have immunomodulatory function, an increase in the fasting group compared to the controls, and very interesting, there was a very strong correlation of the abundance of Faecalibacterium with levels of leptin in the serum. By looking at the gut microbiota, interesting, we compared the changes that we have observed in the EAE studies compared to what we have observed in the human study by looking at the four major phyla, which are the same in mouse and humans, even if the structure of the gut microbiota is profoundly different. And we have observed that the changes in these four phyla were going in the similar direction in the mouse and in the human study, with an increase in the Firmicutes with fasting, while a decrease in the Verrucomicrobial and Lactobacteria. We also did immunophenotyping, extensive immunophenotyping in the peripheral blood by flow cytometry. And what we observed is that in the control group we had an increase over all of the different subset of lymphocytes, which was probably related to the treatment with steroids. And interesting, with the group that was on fasting did not show this marked increase, which resulted in a significant difference on day 15 for several of the lymphocyte subsets. So definitely we were able to modulate the number of immune cells, especially lymphocytes, with fasting. We also looked specifically at the effects of the diet on regulatory T cells. So absolute counts of circulating Tregs were unchanged between the two groups at day 15. However, in functional studies in vitro, Tregs isolated from the fasting group were more suppressive on the proliferation of T effector cells in vitro compared to those isolated from the ad libitum group. On the left, you can see the graph in which we analyze the suppressive activity at baseline and the suppressive activity of Tregs on T effector cell proliferation, and at the different ratio between the two cell types. We do not see difference between the two groups, while at day 15 the suppressive activity of the Tregs from the group on intermittent fasting was maintained higher compared to the control group. So, in summary, what I've shown is that there are pretty consistent evidence from the literature that obesity in children and young adults is a risk factor for MS. And this is associated, and obesity is associated with increased systemic inflammation, which can contribute to autoimmunity. In our study, we have shown that chronic and intermittent calorie restriction have anti-inflammatory effect and then can ameliorate EAE through the modulation of circulating hormones and adipokines, as well as effect on the gut microbiota that could participate in the beneficial effect of calorie restriction, as we have shown in fecal microbiota transplantation experiments. We have also performed a pilot trial of intermittent energy restriction, relapsing remitting MS, which showed that intermittent energy restriction is well tolerated. There was a good compliance and no safety concern based on routine testing. We have seen that leptin levels in the serum were significantly reduced in the fasting group compared to the control, while we did not see changes in serum adiponectin and beta-hydroxybutyrate. While we saw an effect in peripheral immune cells, the different lymphocyte subset, as well as the function of regulatory T cells. The main limitation of our study in people with MS are the small sample size, the short duration, and of course the inability to blind patients to the diet assignment, which is a problem of all the diet study, and also the confounding effect of the concomitant treatment with the corticosteroid for the relapse. In order to overcome, at least in part, this limitation, we are now doing a larger and longer study to test the effects of intermittent fasting in patients with relapsing remitting MS, which are stable and not relapsing. The study is funded by the National MS Society and it's currently ongoing. Unfortunately, the enrollment was delayed by the ongoing pandemic, but we are close and we hope, by next month to complete the enrollment. In the study, we are enrolling 40 people with relapsing remitting MS, either untreated or on some disease-modified treatment. They will be randomized to either fasting, which will be intermittent energy restriction, as we did in the previous trial, for 2 days per week. The intervention will last 12 weeks, and the primary outcome is also mainly changes in adipokines, but we are also always collecting stool samples for gut microbiome analysis. We are also looking at several other parameters in the blood, as well as we are performing DEXA to study changes in body composition, oral glucose tolerance test, as well as I was delighted to be able to add for the last 10 subjects enrolled also brain MRI. And finally, I would like to thank all the people that are involved in this study, all the members of my labs at Washington University in St. Louis, Dr. Cross, who is the head of the Neuroimmunology Division, Dr. Amber Salter, she's the biostatistician helping us with all the studies, Dr. Luigi Fontana and his group, who assisted us with all the human studies, Dr. Injiao Zhu, who performed all the gut microbiome analysis and all funding for this project, and finally, thank you also for your attention.